Picture this headline: Decreased milk production in cattle due to parasitic outbreak. What if I told you that the key to this problem could be found in microscopic markers in blood cells? Incredible, isn't it? We have spent the last 3 months analyzing molecules invisible to the human eye to learn the distinctions between human and bovine red blood cells after stress exposure. My name is Sonia Mirza, and today I would like to share with you the big ideas behind tiny cells. So the cells in question are the red ones. They're responsible for transporting oxygen throughout our body, and they also maintain our pH levels thanks to their shape and the heme protein, which does a bunch of gas exchanging. Simply put, they keep us alive. Over the years, we have accumulated so much data about our own red blood cells, enough for us to be able to treat disorders like anemia. Under normal circumstances, our blood cells live around 120 days, but with anemia, this lifespan is cut short. Factors like oxidative stress, a lack of glucose, or high body temperatures can all lead to premature cell death, or eryptosis. Now, what about the cells of other mammalian species? Well, we are intertwined with the environment around us, so the well-being of other organisms will work to affect us. Due to this, we chose to analyze cows. Being a species that we consume and one that we so often take care of, it is in our best interest to know as much as possible, and right now, a lot is unknown. If we let our cattle be affected by diseases like anemia, we will be met with lower dairy production and just overall weakened cows. So to mimic the oxidative stress that can occur in the body, we use a chemical known as tert-butyl hydrogen peroxide. Try saying that 5 times fast. These samples were then stained with a protein dye called annexin 5, which binds to a specific molecule known as phosphatidylserine, and you can see this on the slide. Phosphatidylserine is typically found on the inside of our cell membrane, but due to eryptosis, it flips outward, allowing our stain to latch on and show us how many dead cells there are. Post-oxidation, we saw a lot of dead cells. The same was applied with indirect oxidative stress, which we achieved by exposing our samples to high temperatures and in another test, depriving them of their energy supply. We then use a compound called DCFDA, and I'll spare you the tongue twister here, which binds to unstable ions known as free radicals, an indication of oxidative stress. And as you can guess, a lot of free radicals were present in our treated samples. But that's the problem. Too many free radicals cannot be handled by the body. After a certain point, these cells will start to undergo oxidative damage. If we let our cattle be exposed to parasitic diseases that cause fevers, or if they're not receiving enough energy in their diet, they will undergo oxidative damage and struggle to produce as much milk. Our examination of these tiny markers within bovine blood allowed us to get a clearer picture of what affects bovine health and milk production, and we can continue to learn more so that they can thrive under our care. Thank you.